All right, so we've talked previously about what DNA looks like, how DNA is replicated, but DNA ultimately is the instruction manual to build proteins. And so today we're going to talk about how those proteins are made from the instructions provided on by DNA. So the biggest thing is for you to understand that in order to become a protein, DNA gets transcribed into RNA, specifically mRNA, and then that gets translated into a protein. So it's a two-step process where DNA becomes, or it's transcribed or transcription into RNA, and then translated to protein. So transcription, what is it? It's the copying of DNA into a complementary strand of RNA. Transcription occurs in the nucleus. So what's the difference between DNA versus RNA? So where DNA starts with a D, it's deoxyribose. RNA is also a sugar, but just ribose. And deoxy just means it's without two oxygen. Deoxy, so D meaning without, oxy referring to the oxygen. DNA is double-stranded. RNA has a single strand. And then the base pairs are a little different, where we have adenine, thymine, cytosine, and guanine. RNA shares adenine, cytosine, and guanine, but it has a U. Instead of T, it has uracil. So RNA, instead of thymine, has uracil. There's different types of DNA. DNA can be found in nucleus, mitochondria, or chloroplast. There's three types of RNA as well. mRNA, which is messenger RNA, tRNA, transfer RNA, and rRNA, which is ribosomal RNA. DNA never leaves the nucleus. RNA is, during transcription, it stays in the nucleus, but then when it does translation, it moves out of the nucleus into the cytoplasm, where it finds a ribosome. So this is what DNA and RNA looks like. You can see RNA on the left here. Notice it's single-stranded. Notice that it has CGA, but it has U, whereas DNA has the T. What happens during transcription? So what happens is in step number 2, where we attach nucleotides in DNA replication, use DNA polymerase. Well, use RNA polymerase. And so what basically it does is it's similar to step number 2, where the RNA polymerase, it unzips the DNA, and then here's what we call the template strand. So this is the DNA template strand. And what it does is it attaches RNA, so ribonucleotides, just like step number 2 of the DNA replication, but instead of making a complementary DNA strand, it makes an mRNA strand. M, if you remember, stands for messenger. And you can think of it, it's the messenger between DNA to proteins, right? So that's transcription. So transcription is making an mRNA strand. So if we wanted to practice, if we want to make mRNA, the mRNA strand, so the RNA polymerase would spot the G and would put a C. It would spot the A, and normally it would put a T, but remember, on RNA there is no T, so we put a U, and then you see the T, and they put an A, C goes to G, G goes to C, G, C, A. There's no T, so you put a U. So the U replaces the T. Okay. And so that's mRNA. So now, after transcription is done, you have a piece of mRNA, which is complementary to the DNA template strand. And so it's hanging out in the nucleus because transcription occurs in the nucleus. And now we're going to go to do translation. So what is translation? It's the process of making polypeptides or proteins. And so the mRNA, what it does is it leaves the nucleus, goes into the cytoplasm. And so the code of the mRNA is read in threes. So like 3 base pairs equals one amino acid. And so it might be easier to see. So what happens, let me see if there's a better, this is the best one here. So here's the mRNA strand, right? It was just made after transcription. And what happens is you have these, and these are called tRNA. And the tRNA have 2 things. They have this part, which we call an anticodon. Why? Because this 3-section interval is called the codon. So the anticodon, notice that the C will pair up with the G, this G will pair up, so it brings this. Now, on its tail, so to speak, it has the protein, it has the amino acid. So this tRNA carries an anticodon and an amino acid. So when it hooks up with the correct codon, it'll bring along an amino acid with it. And so what happens is as more tRNAs arrive, what they'll do is they'll start building a like a protein, an amino acid chain, which ends up being a protein. Because proteins are just a polymer or multiple amino acids bonded together. So just to recap, this is your mRNA strand. They're separated in codons. And so the codons are 3. So like here's a codon, and then there was a codon here. And what happens is the tRNA brings the corresponding codon, or the anticodon, and with it, it carries an amino acid. So amino acids, they code for 64 combinations. If what you do is, since you have 4 possibilities, if you multiply 4 by 4 by 4, you get 64. So there's 64 possibilities, right? So we have like a codon chart, and this is the this is the mRNA. So, for example, if I had a UAC, so if I wanted to know, this is a codon on the mRNA. If I wanted to know what it coded for, or which amino acid, I could use the codon chart. So in the first position, I have a U, so U. Second position, I have an A, so here's A. So it's, right now we figured out it's here, and then C is here. So if I look at C, I see that this codes for tyrosine. So the tRNA would bring a tyrosine along. So that would be my first one. Okay, so this is how you use the codon chart. This is another example of a codon chart. So here we have an example. Let me do this. Let's see if it's big enough. Let's see if I can. This might be a little too hard to see. Let me remove this. Okay, let's see if this works. So let's do this example together. So first we have our DNA. So it gets transcribed into mRNA. So C-U-A-G-C, A goes with U, and then A. All right, and then what we're going to do is we're going to do the amino acid sequence. So we start with CUA. So C, sorry, first position, C. Now you notice that it'll be proline no matter what. So, but C, I'm sorry, C, U, so that's leucine, and then A. So it's leucine, which is like this. Now you can, there's abbreviation, so you could do LEU. What's the next one? So what's GCG? So G is down here. Let's do this real quick. I'm going to go ahead and get out of it. Let's try it one more time. So this was CUA, GCG, CUA. So CUA, we agreed, was leucine. GCG, here's G, here's C, and so it's going to be alanine. And this is CUA, so CUA, oh, that's the same thing, isn't it? So it's going to be leucine. Okay, so, and then this, if you put a bunch of these together, these amino acids, you'll form a protein. And so that's how we make protein. And what you'll see is it's based off of the mRNA, which is based off of the DNA. So the DNA ultimately produces, or is the instruction manual for the protein synthesis, the making of protein. All right, oops. All right, let's leave it at that.